Looks like you got one of those nice standing desks, Maggie.
Yeah, that was something I had to that I got when I realized I'd be spending a lot of time in my home office, which is the size of a shoebox, by the way.
I will say, like, they did get quite popular at the Smithsonian, like pre-COVID. They kind of like started spreading around the Smithsonian as a popular thing to get. So
yeah, that was one of the first ones that I tried. I was like, I could do this. It's not so bad.
Yeah.
You just change your perspective a little bit. Like, you know, if you're like getting a little sleepy, stand up.
Stand up, get the blood flowing.
Oh, I see some of my students in marine conservation class. Great. Nice. I'm proud of you.
Someone is asking in the chat, Jeremy.
Yeah, so it will be recorded. Yeah, it will be recorded, and it will be available on the website of the Institute of Environment. So for those, I mean, we have all the seminars, the marine science seminars on the website.
Okay. Yeah. How do you usually field questions? You do that at the end, just people chime in with questions?
Yeah, it's usually small enough that people can just unmute their mics and ask questions. We haven't really been using the chat, but people can type in questions into the chat. I guess even if you have questions during the seminar, you can type them into the chat. Maybe we can cover them afterwards. So yeah, we'll see how it goes. We'll just kind of play it by ear.
Okay, yeah, that's great.
Okay, 301.
All right, do you want to go ahead and get this started, Jeremy? Should we start?
Let's do it. Yeah, we have 36 people. So.
All right, cool. Okay, everyone. So it's just past 3 p.m. So let's get started. Welcome to the third Marine Science Seminar of the semester. Jeremy and myself are excited to have everyone join. For those who are new, we're trying to host these seminars about once a month on Wednesdays from about 3 to 4 p.m. So without delay, I have the pleasure of introducing today's speaker, Dr. Maggie Johnson. Dr. Johnson is a postdoctoral scholar in the biology department at the Woods Hole Oceanographic Institution. She studies how local and global environmental changes affect valuable ecosystems such as coral and oyster reefs. Very broadly, her research pairs in situ environmental and community observations with controlled laboratory experiments to understand, one, the effects of ocean acidification, warming, and nutrient pollution on the ecophysiology of foundation species. Two, the consequences of environmental change on the community structure of marine habitats. And the topic of today's seminar, three, the role of ocean deoxygenation on the functioning and persistence of coral reefs. She has conducted research across a wide variety of locations that would make anyone jealous, from Panama to Hawaii to the remote islands of French Polynesia. Dr. Johnson completed her master's degree at California State University, Northridge, and her PhD at the Scripps Institution of Oceanography at UCSD. She has also completed several postdoctoral appointments at the Smithsonian, and in 2018, she received the prestigious Marie Curie Fellowship from the European Union, which she subsequently had to decline in order to accept her current position at Woods Hole. She has received over $1 million in funding from a diverse array of sources, most recently serving as a co-PI on a large NOAA coastal hypoxia research grant. Lastly, Dr. Johnson has a total of 20 publications in top journals such as Trends in Ecology and Evolution, Nature Communications, Current Biology, and Coral Reefs. Today, Dr. Johnson will be discussing ocean deoxygenation, an emerging stressor on coral reefs. So with that, please join me in welcoming our speaker. And Maggie, I will turn it over to you.
Okay, that's great. Thanks so much for the introduction, Justin, and a big thank you to Justin and Jeremy for the invitation to speak with you all today. It's a pleasure to be able to talk with you a little bit about some of the research I've been doing lately on ocean deoxygenation, which is honestly something we don't typically think about in the context of coral reefs. So that's something that I hope you take away from this talk today, is that we're now considering ocean deoxygenation as a really key emerging stressor for coral reefs. So we all know that coral reefs are important and valuable ecosystems for a variety of reasons. They're built by foundation species, the corals, that create the three-dimensional structure and framework for the habitat, like you can see pictured in this photograph here, which is showing a healthy, vibrant, thriving coral reef ecosystem from the tropical Central Pacific. And the production of calcium carbonate and creation of the habitat by corals is really important for supporting up to 25% of marine biodiversity on coral reefs, even though coral reefs cover less than 1% of the seafloor. They also provide a variety of other important ecosystem goods and services, and by supporting fisheries, coral reefs can provide protein for up to 1 billion people in coastal communities. And that three-dimensional structure that reefs provide is really important for protecting the shoreline of coastal communities, protecting them from damaging activity of waves and storms. And then we have the other aspect of coral reefs, which is just the inherent beauty and cultural importance of coral reefs. Now, unfortunately, what we're getting used to seeing is not those thriving, beautiful coral reef habitats, but instead things that look a little bit more like this. So massive degradation on coral reefs, coral bleaching events, so much so that this has now reached mainstream and is something that, for example, my parents are reading about in the news. And the main goal of my research is trying to understand what the drivers of degradation are on coral reefs. So how we can go from a Caribbean reef that looks like this, where you have live coral cover and a somewhat thriving ecosystem, to within a matter of days, an ecosystem that looks like this. And this is literally a photograph of the same habitat from a reef I work on in Caribbean Panama, where within a few days, we saw the reef transition from a relatively healthy, happy habitat to one that was largely devoid of living coral tissue. And so that brings us to the topic of today, and that's ocean deoxygenation. And really this question of how ocean deoxygenation is contributing to the decline of coral reefs across the planet. So I typically think about the impacts on coral reefs in two categories. So much of the reason that we're seeing decline on coral reefs is because of anthropogenic impacts, so what we're doing to the environment. And we can break those impacts into two categories. So we have our local stressors or our local impacts. And those are things that are happening at the local scale where you can see the source of the stressor. So for example, that includes sedimentation, nutrient pollution, overfishing. And that's in contrast to our global stressors, which are related to increasing carbon dioxide emissions in the atmosphere. And those are things like warming and ocean acidification. Now, what's important to remember is that our global stressors, like warming and ocean acidification, are not occurring in a vacuum, that they're actually occurring on this landscape or this background of what's happening at the local scale. And so really it's this interaction of local stressors and global stressors that influence the outcome of those impacts on an ecosystem. And so how things are going to manifest in your local habitat or your system that you're working in from the global scale is really going to depend on what's happening at the local scale. And one interesting way to think about this and to think about the interactions or potential interactions of local and global stressors is by studying deoxygenation, which I will also use interchangeably with the term hypoxia. So deoxygenation is referring to the loss of oxygen from our global oceans. And oftentimes we think about deoxygenation as ocean deoxygenation in the open ocean, oxygen minimum zones, or we might think about something like coastal hypoxia and mass mortality and formation of dead zones, such as we see in the Gulf of Mexico. But really these two processes are basically driven by the same fundamental process of the loss of oxygen from our surface waters, which is why we've made a transition towards referring to this process as deoxygenation altogether. And so to explain what I mean by this interaction of local and global stressors and how that amplifies hypoxia and deoxygenation, I'm going to show you this figure here, this schematic. So think about the coastal ecosystem, your favorite one, whichever you like to think about. So I'll think about a coastal coral reef, but this could apply to something like a seagrass or your salt marshes, any sort of subtidal habitat. So we have nutrients that go into that habitat. And this is something that's happening at a local scale. So we have eutrophication or nutrient inputs that can increase algal productivity in our coastal habitat. And what that's going to do is it's going to fuel algal productivity and can cause the formation of algal blooms. And that's what this little cloud here is symbolizing. At the local scale, we can also see things like pollution that can increase organic inputs and increase heterotrophy in the system, which is going to be consuming oxygen. Now you couple that with what's happening at the global scale, this would refer to things like warming. So warm water holds less dissolved oxygen. So as temperatures increase, seawater holds less oxygen. But we also see that warming increases algal growth and it can increase stratification. And that's what's depicted in this diagram here. So we have our algal bloom that's being fueled by higher nutrient inputs. We see warming causing increased stratification where we have the saltier, higher density water down here at the bottom. Because of that stratification, what we can see is low oxygen concentrations right along the benthos that are caused by heterotrophic respiration of bacteria and other microbes. And if these conditions continue, then we can see this hypoxic area, this area of low oxygen concentration, grow and grow and grow and get bigger. And what we're seeing is an increase in the frequency and the severity of these hypoxic regions or the formation of hypoxic events across the globe. Now, deoxygenation isn't something that's necessarily new per se, but it is something that we're thinking about sort of newly in the context of coral reefs. And before I get into the specifics of some of the experiments that I've been doing, I want to give you a little bit of a background about some of the numbers because I will be referring to hypoxia and normoxia. So first off, hypoxia is referring to lower concentrations of dissolved oxygen in water. And different levels of hypoxia can alter organismal behavior in different ways. So when conditions are what we'll say normal or normoxic, this is when the amount of dissolved oxygen in seawater is around 6 to 8 milligrams per liter. And that's going to vary a little bit depending on temperature and where in the world you are. This is where we see normal behavior. We then have hypoxia, which it depends a little bit on what organism you're talking about, but very generally hypoxia typically refers to when those oxygen concentrations decrease to about 2 to 3 milligrams per liter. This is where we start to see behavioral and physiological effects. So things like fish kills, mass mortalities, when you have critters that can't escape from that low oxygen water. And then at 0 milligrams per liter, this is where we have anoxia, that's zero oxygen in the water. And this is where we see anaerobic processes. We do not see any aerobic processes. And so keep these concentrations in mind once we get down the line a little bit when I'm telling you about some of the experiments. So deoxygenation, again, hypoxia, we've known for a while that this is a really important process, particularly in temperate ecosystems and coastal seas. And we know that the loss of oxygen can cause dead zones, mass mortalities, and really ecosystem collapse in these ecosystems. So it's not necessarily a new topic, but what is new is considering ocean deoxygenation in the context of coral reefs, because for the most part, coral reefs have been largely ignored with respect to deoxygenation. And so this really first came to more general awareness in this paper by one of my postdoctoral advisors, Andrew Altieri. It was published in 2017, documenting for the first time a really comprehensive documentation of hypoxia on a coral reef. In addition to that, we conducted a study looking at where dead zones and hypoxia events have been documented in the world. And so on this global map, these red dots here are showing where there has been a documented dead zone. The orange dots are showing you where hypoxia has been implicated in mortality on a coral reef. And just looking at the graph, you can see that there are a lot of red dots. So we know that dead zones have been documented in a lot of places, but not very many in the tropics. And that's not because there isn't a lot of habitat. All of these purple regions are showing you where we have potential coral reef habitat. But there's a really large gap in our knowledge of what hypoxia means in the tropics, how often it occurs, and what it does to the ecosystem. And so this is a gap that we are trying to fill right now. And it started with some of Andrew's work documenting that first hypoxic event in Bocas del Toro, Panama, that occurred in 2010. And so during that event, so 2010, what you're looking at here is a map of the Bay of Almirante. So here we are in Panama on the Caribbean coast, kind of near Costa Rica. And they documented oxygen concentrations in the water column and on the benthos during a hypoxic event. What I want you to take away from this is that we have in this red color here, oxygen concentrations that went down to near-anoxia on a shallow coral reef. The take-home message from this first study was that as you decrease oxygen concentrations during a hypoxic event, you see a significant decrease in the amount of live coral cover and in coral species richness. And that makes sense. So when you decrease oxygen concentrations, and those conditions persist for a certain period of time, we can see mass mortalities in those regions. But what's really interesting about this first set of work is that not all corals died, and it seemed that some species appeared to be more tolerant to hypoxic stress than others. And this is a theme that you'll hear me talking about as we go through the rest of the talk, is trying to understand why some species are more tolerant and which ones are more tolerant, because this has really important implications for the future of our coral reefs. So to get into the work that I'll talk to you about today, what we're going to be thinking about is how does ocean deoxygenation contribute to reef degradation? And for my talk today, I have two objectives. The first is to tell you about an in situ hypoxia event on a coral reef. And this is building a little bit off of that Altieri study that I showed you from an event in 2010. But this is one that occurred in 2017. And then second, to tell you about how we're doing controlled laboratory experiments to try and identify the specific role of oxygen in coral responses during these hypoxia events. And so first, we're going to travel to Bocas del Toro, Panama, where I was based for my first postdoc, and I lived in Bocas del Toro for about a year. And toward the end of that time, we detected another hypoxia event on the coral reef. So this is 2017, 10 years after that first event occurred. And this is a project that I've been working on with my collaborators and postdocs at STRI and one of your very own students, Will Weed, who was involved with the project while he was working at STRI in Bocas del Toro. So there's some interesting things about this region that make it predisposed to the formation of hypoxic conditions. So first detected, really documented in 2010, and then we see this happen again in 2017. So what's going on? What you're looking at here, so you've got the map of the Bocas del Toro region. The yellow star is showing you where the STRI Bocas del Toro research station is for context for those of you who are familiar with it. So we found that this region seems to be at increased risk of hypoxia, and the potential reasons that this may be is because, one, human impacts. There's a lot of eutrophication in this region from banana plantations, and unfortunately, there's also quite a bit of organic pollution because there's a lot of raw sewage effluent that goes into the bay. And then on top of that, in this bay region in here, it's relatively reduced circulation because there are really only two sources of inflowing water to the system. One is here and the other is over here. So there's reduced water circulation, particularly in the farther reaches of the bay back here. During periods of low wind activity and really warm temperatures like we see in the summer, we can see some really intense stratification forming back here in the farther reaches of the bay. And this is typically when we start to notice those hypoxic regions on the benthos growing, and that they can then show up onto the coral reefs and cause them pretty serious problems. Okay, so 2017, September, this is what it looked like at the surface at one of my sites. Absolutely beautiful, flat, calm, clear waters, gorgeous day. And when we got in the water and we looked below, the case was not the same. So what you're looking at here is what we saw on that day in 2017 when we first detected this event. You're looking at a mounding coral that has a lot of invertebrates that have climbed to the top of the coral to try and escape low oxygen conditions in the seawater. And so, for example, you have these brittle stars that are normally cryptic that you would not normally see out in the day. We have all these invertebrates that were just piled up on top of each other, which is one of the kind of characteristic defining features of a hypoxia event. So they're low oxygen concentrations. The organisms that can move are trying to flee and get away from those low oxygen concentrations. So we also saw lots of mortality of sponges and mass bleaching of corals that were attached to the benthos. So for the brittle stars or the crabs or the fish, they can swim away from those hypoxic waters. But for the things that are attached to the bottom, they can't escape. And so we saw a lot of necrotic sponge tissue. Here you can see this is a sponge that has died and now has a microbial mat growing over the surface of it. You can see that there's some bleaching of the coral in the background here. And this is a massive mounding coral that's, probably twice the size of me, that over, a couple of days was totally bleached. We see invertebrates piled up on top of each other at the top of those coral heads, as high in the water column as they could get. We've got our brittle stars, we have lots of little snails. And so after we detected that this event was occurring, we went out and we measured the oxygen concentrations in the water column. So one of the tricky things about these hypoxic events is that they happen really quickly. A lot of times we don't know when it's going to happen. So you have to respond quickly and try to capture the event while it's in progress. And to date, this hasn't really been done very well for a coral reef. So it hasn't been done comprehensively where we have the environmental conditions and the biological response. So for this study in 2017, we went out, we measured the dissolved oxygen concentrations in the water column and on the benthos. And that's what you're looking at here. So again, during this event, we saw formation of hypoxic to near-anoxic conditions on the benthos of the coral reefs in this region. Across this gradient of dissolved oxygen concentrations from anoxic to normoxic, we selected two sites. One here, that was from the pictures that I showed you before, where we actually detected the event. And then we chose a site that was far outside the bay and was constantly flushed with seawater. And so we knew that it had normoxic conditions. And we compared the corals from those two different sites to try and detect the physiological response of corals during an actual hypoxic event. And, looking around during the event, you could tell that there were corals bleaching, but we wanted to be able to actually put a number on it and to quantify it. And so we focused on a common Caribbean coral, Agaricea tenuifolia. We collected it from each of those sites, and then we measured the photophysiological response. So this is specifically, we're looking at maximum quantum yield. And this is a metric that I'll tell you about a little bit more, so I'll take just a minute to explain it. So remember that corals have endosymbiotic algae living inside their tissues. And so photosynthesis is a really important biological process for maintaining healthy corals. And that intact symbiotic relationship is really important. When corals get stressed, that symbiotic relationship breaks down. And this is something that we can detect by looking at photophysiology. So as that relationship breaks down, photophysiology of the coral or the symbionts that are associated decreases. And so changes in that photophysiology can tell us if a coral is starting to bleach and it's starting to be stressed. So in addition, we also looked at symbiont densities and we looked at chlorophyll A content, which tell us a little bit about how many symbionts are in the corals and how they're doing with respect to photosynthesis. So here what you're looking at is maximum quantum yield at the normoxic site in blue and at the hypoxic site in red. And this is our measure of essentially photosynthetic efficiency or the health of the coral you could think about. And so what was clear is that at the hypoxic site during that event, maximum quantum yield of those corals that we sampled was lower, significantly lower than the ones sampled at the normoxic site. And this matches what we could visually detect. So here's a photo of some of those corals during the event. And we found the same sort of patterns in their symbiont densities and their chlorophyll A content. So what this tells us is that we have physiological data supporting the fact that corals are bleaching during these hypoxic events. That you can see here. Okay, so that's the physiological response. So now what we want to know is, how does that physiological response translate to a community response? What does that mean at the community level? So at each of the sites that we surveyed, I had a series of permanent photoquadrats that I've been monitoring that we could use to see how the community was changing before, during, and a year after that hypoxic event. And so what I can tell you is that before the event, live coral cover at our hypoxic site, where the event occurred, was around 30%. And what you're looking at here, y-axis, is percent cover of these dominant functional groups. And so our live coral is in that pinkish color, dead corals in the red, and those are the ones that I'll primarily be talking about. Now, during the event, that live coral cover decreased by about 50%. Notably, we saw an increase in bleached corals, partially bleached corals, and in dead corals. And then 1 year later, so this is after about a year period of recovery, we saw that live coral cover was about 25% lower than when before the event had actually occurred, but there were also more dead corals. Now, this is particularly interesting because in the 2010 event that Andrew Altieri did with his colleagues, they found near 100% mortality of corals on the benthos, which means that a year later, those corals were still dead. So here what we're finding is that the shallow coral reef community that we surveyed was actually more resilient than the deeper community. But we did see changes to the structure of that community that persisted even a year after the event. Now, to give you an idea for what that looks like a little bit, what that community change looks like, this is 2017 during the event. This is a thicket of Acropora silvicornis. Unfortunately, this is one of the last living Acropora silvicornis thickets inside the bay in this region. This is a year after the event. So the corals died during the event. This is dead coral, basically dead coral thicket, no living tissue. And then without that living coral tissue, you see breakdown of that Acropora thicket and complete loss. So that's a really big change. And sometimes that visual impact is, or that visual is a lot more impactful than just talking about the numbers. What we've seen since that event occurred in 2017 is that recovery of the community has been extremely slow. And this is that hypoxic site 3 years after the event occurred in 2017. And so while we did find that some corals survived the event, they were typically up more shallow than the ones that you see down here. So these massive boulder corals bleached and died during the hypoxic event and will not ever really recover. What we're seeing is some recruitment back into the system, but for the most part, the structure and function of this ecosystem has been completely altered from that hypoxic event. And just a reminder, this is what it looked like before the event. Okay, so that was the first objective, to characterize an in situ hypoxic event more comprehensively, so to characterize the environmental characteristics, as well as the biological responses of organisms in the community. And what we found was that during the event, residents of that community were exposed to near-anoxic conditions for prolonged periods. Now, we don't know exactly how long those conditions lasted, but we know that it was long enough to have a long-lasting impact on the benthic assemblage. Corals suffered bleaching and mortality, but key is that not all of them did. And so this is an idea that we'll explore in the next section of my talk. And what we see from this and from the previous events that we've detected is that hypoxic events can decrease live coral cover from anywhere from 50 to 100%. And that recovery, particularly in the system that we've been working in, is extremely slow. And so this tells us that these events can occur in the tropics on some reefs, and importantly, that they have really catastrophic impacts on the community as a whole. So the next step is to think a little bit more about what oxygen stress itself means for corals. So the tricky thing about studying these in situ events is that there are a lot of things that co-occur at the same time during a hypoxic event. And so I'm very careful to say that these are responses to the event itself, and we don't know exactly how much hypoxia is contributing to that response. So for example, during hypoxia events, we typically have warmer water temperatures, sometimes we have lower pH, and sometimes we also have higher chlorophyll A content. But I will also say that the magnitude of change in those other factors is nowhere near the change that we see in dissolved oxygen concentrations. So the next step is to really try to identify what the specific role of oxygen is in coral bleaching responses to things like hypoxia. And the way that we can do this is by using controlled and targeted experiments where we manipulate oxygen concentrations and look at the effects on different species of coral. And so that brings us to the second part, where we're going to be thinking about what oxygen stress by itself means for the response of some different reef building corals. Now, this work is part of a grant that I'm involved with my co-PIs, Andrew Altieri and Valerie Paul. This is through NOAA's Coastal Hypoxia Research Program, where we're assessing the threat of hypoxia to tropical ecosystems in a restoration context. And I'll talk about this restoration context a little bit in just a minute. So these are experiments that we've been conducting at the Smithsonian Marine Station in Fort Pierce, Florida, which is where I was based for the second part of that postdoctoral fellowship that I mentioned. We also have a PhD student from the University of Florida, Sari Swaminathan, who's been working with us on this project and has been hugely instrumental in getting some of these experiments done. So to give you an idea for what these experiments look like, it's very similar to ocean acidification experiments for those of you who have experience with that. So the idea is that we want to simulate deoxygenation in the lab. And a way that we can do that is by bubbling in nitrogen gas, which is symbolized by this reddish orange tank here, using that nitrogen gas to drive down oxygen concentrations in seawater in our replicated tanks. And then we use air for our ambient controls for our normoxic conditions. And using this approach, so that we used a feedback system using dissolved oxygen probes in each one of these tanks, we're able to create a range of deoxygenation treatments, going from 0.5 milligrams per liter of dissolved oxygen all the way up to ambient or normoxic conditions, which were around 6 milligrams per liter. And to give you an idea for a little bit of this output, this is from one of our experiments that we ran for, it actually ran for about 14 consecutive days, but I'm showing you 11 days of data here. This is showing you the hourly dissolved oxygen concentration for specific treatments. So we have the 0.5 treatment, 2, 4, and 6 milligrams per liter represented by these lines here. So we had some issues on this day here where we ran out of some nitrogen gas, but we were able to replace that. But this is a really good test for our ability to maintain continuous near-anoxic conditions for our corals to grow in. So the goal of this experiment is to explore the role of hypoxia in coral responses. So looking at coral condition and photophysiology as metrics for how the coral itself is doing. And specifically, we want to know about how 2 different species of coral that are used in restoration efforts throughout the Florida Keys, how they respond to those hypoxic conditions. And our target species, the first one is the Cropper cervicornis, or the staghorn coral, that you can see pictured here. And this is one of the fragments that we used in our experiments. And then Orbicella fabulata that you can see pictured here. So again, these are 2 species of coral that are used in broad restoration efforts throughout the Florida Keys, as well as the Caribbean. And so understanding how these species respond to another potentially important stressor is really important for identifying where these species should be outplanted or where they should be used in restoration efforts. So what we did was we exposed each of these coral species to those 4 hypoxic or 4 oxygen treatments that I showed you before. We measured their photophysiological response, much like I explained for the field experiment that we did. We looked at tissue loss and mortality for the duration of the experiment. And we also looked at symbiont densities at the end. Now, the nice thing about using the PAM fluorometer for photophysiology measurements is that it's non-invasive. So we can actually measure coral responses over time without having to do any destructive sampling. For tissue loss, we evaluated each of the fragments on a daily basis to quantify % of the fragment that showed any signs of tissue loss or mortality. And so first we'll take a look at those photophysiology responses. So here what you're looking at is quantum yield on the y-axis. So this is essentially the photosynthetic efficiency of the coral. And remember that decreasing quantum yield is telling us that the corals are a little bit more stressed and either bleached or on their way to bleaching. We have time on the x-axis from the first day of the experiment all the way to the 5th day. So here we have, in the top set of figures, you're going to see the results for Acropora cervicornis. And then we have our color-coded treatments, and we have 2 different measures of quantum yield. We have maximum quantum yield, which was measured at dawn. So this is after the coral had been in the dark, not photosynthesizing for a full night cycle. And then we have effective quantum yield, which is when we're measuring the photophysiological response at peak midday when the coral is photosynthesizing. And I mention this because we see some pretty interesting responses depending on what time of day we took measurements. Okay, so for Acropora, what we see is that at around day 3, our maximum quantum yield starts to significantly decline, but only in that most extreme treatment of 0.5 milligrams per liter. We only measured effective quantum yield on 2 days for Acropora. We see that after 4 days, we also see a significant decline in that quantum yield in the most extreme treatment. But notably, our intermediate treatments are no different than the ambient treatment, which is pretty interesting. Now, what's really interesting is when we look at the response of Orbicella over time. So what you'll first notice, so the graph is set up the same way, but you'll notice that the experiment goes to about 11 days. And this was a little bit tricky. So we were trying to decide the best time to end the experiment because we have some destructive sampling that we needed to do where we actually needed to have living coral tissue. So for example, we couldn't have all of our corals die because we needed to be able to sample the tissue for zooxanthellae densities and things like that. And so we decided on ending the experiments when half of the corals in the lowest treatment, in the 0.5 treatment, showed signs of mortality or partial mortality. With Orbicella fabulata, we saw some decrease in quantum yield at around day 8, but that was only during that early dawn measurement. So after the corals had been in the dark all night long. For the measurements that were taken midday, we didn't see any effect of those oxygen treatments on the photosynthetic response, which is pretty interesting. So some takeaways from this first set of data is that we really only saw effects of extreme hypoxia on these 2 species of coral. We really only detected a response in Acropora after about 3 days, and there were minimal to no effects of Orbicella after 11 days. So what I'll say is that at 11 days, we decided to conclude the experiment because keeping corals in prolonged hypoxic conditions for more than 2 weeks doesn't seem necessarily ecologically relevant. And also it's extremely time consuming to run these experiments. So we cut the experiments off after we found that they weren't really responding to treatments. Okay, so tissue loss and mortality. First off, there was no tissue loss or mortality in any of the Orbicella fragments. And so what I'm showing you here is a figure of tissue loss for Acropora only. And that's from the period of the beginning of the experiment to the end. And what we saw was a significant increase in tissue loss in the most extreme treatment. We see a little bit of tissue loss in the second most extreme, so 2 milligrams per liter, but no effect in our ambient or our near ambient hypoxic treatment. So again, so tissue loss in Acropora started at around day 2. So this is a really quick response. We only saw tissue loss and mortality in Acropora, nothing in our Orbicellas. And to give you an idea for what this looks like when I'm talking about tissue loss, it's really obvious in these corals. So what happens is, let's see, here we have our ambient treatment for Acropora cervicornis. We have Orbicella fabulata. We start out with the initial, and then we have day 3 or day 7 for the midpoint of these experiments, and day 4 and day 14. In Acropora, what we can see is these white patches where tissue has started to slough away from the skeleton. And so it's really clear when we start to see this tissue loss in Orbicella. In Acropora. In Orbicella, we saw absolutely no tissue loss, and we saw minimal responses of their photophysiology as well, which tells us that it's not necessarily something that we were missing. It's just they don't really seem to respond that much. So here you can see that really obvious tissue loss in the Acropora fragments. We had about 5 fragments of Acropora that were completely dead by the end of the experiment, but for the most part, most of the fragments had some amount of partial mortality by the time we decided to end the experiment. Okay, so the next step is looking at the symbiont densities, which tells us a little bit about if the corals are bleaching or if they're losing some of that tissue. So here what you're looking at are the dissolved oxygen treatments on the x-axis from 6 to 0.5 milligrams per liter. On the top we have Acropora silvicornis, and we've got our symbiont densities. And so what you see here is at the end, when we sampled our Acropora fragments, so each one of these gray dots is showing you an individual fragment, and the colored dot is showing you the treatment mean, plus or minus the standard error. What we see is that there's a decrease in symbiont densities in the 2 milligram per liter treatment, and the most significant decrease in the most extreme treatment, which matches what we saw in the PAM fluorometry or photophysiology data as well. And when we look at Orbicella, we see no difference across those treatments. And so we do see that our physiological measurements are matching up, where we have strong negative effects of the extreme hypoxia treatment on Acropora, and no effects of any of the oxygen treatments on Orbicella fabulata. So we see significant declines in the symbiont densities in the lowest treatments, but in Acropora only. So what this tells us, this is something that we had suspected based on what we saw in the field, is that corals do demonstrate some species-specific susceptibilities to hypoxia, which is what we would expect based on the responses that we've seen of corals to things like thermal stress or ocean acidification. And they're particularly notable in the 2 species that we used for this study. So for example, I would categorize Acropora silvicornis as highly susceptible to hypoxia. We saw that, the experiment lasted for about 5 days. At 5 days, 50% of the corals in the lowest treatment were either dead or on their way to dying. Orbicella fabulata was tolerant and lasted for, so we had the treatments actually running for 14 days. And we didn't see really any detectable visible responses and minimal responses of their photophysiology. So to summarize and think a little bit more about this, the first objective was to think about in situ hypoxia and what it means for a coral reef. So identifying and bringing awareness to the fact that these hypoxic events are occurring in the tropics and that we don't really know that much about them. So this is definitely an area that should be a focus of future research. From the event in 2017, we found that this hypoxic event induced coral bleaching and caused a shift in the benthic community that has lasted for years. And the second part, we talked about looking at sensitivities of different species of coral to hypoxia alone. So isolating oxygen stress in the lab under controlled conditions and trying to identify how different species respond to those conditions. And what we find is that species respond differently to that hypoxic stress. Orbicella fabulata generally was pretty resistant to continuous exposure to hypoxia. Acropora silvicornis was not, and it was pretty sensitive. And this is a really interesting aspect of the study. So the effects of those hypoxic treatments, particularly in Acropora and even a little bit in Orbicella, were the worst at night. And so this is really important because this can tell us a little bit potentially about why corals are responding the way they did in our experiment. So one thing or one caveat that I'm going to throw into the mix about these experiments is that we manipulated dissolved oxygen concentrations in what I'll call the bulk water column. So we manipulated the concentrations in the water in the tank. We had pumps to make sure that everything was well mixed, and we're confident that the dissolved oxygen concentrations that we had in the tanks was exactly what we were targeting. But what we don't know is what the dissolved oxygen concentrations were in the coral tissues. Okay, so corals have their photosynthetic symbionts photosynthesizing during the day, producing oxygen. So it's very likely that within the boundary layer of the surface of the coral and within the coral tissues, that hypoxic stress is relieved during the day by photosynthesis of their symbionts. Now, at this point, it's not something that we've tested. We would need to incorporate microsensors and look a little bit closer at what's happening in the tissues, but that's likely one potential explanation for the different responses that we saw during the day and during the night, which is pretty interesting to think about. Okay, so what does this all mean? And what does deoxygenation mean for coral reefs? So we know that deoxygenation is a problem on reefs, potentially more than 10% of reefs across the planet, and that's likely an underestimation. So this isn't a problem that is isolated to Bocas del Toro, Panama, where the reefs are kind of funky. This is a problem that is prevalent and increasing in prevalence in reefs across the planet. Some reefs are going to be more susceptible to formation of these hypoxic conditions than others, depending on what's happening at the local scale. Now, with deoxygenation, with hypoxic events, one of the key contributing factors is, for example, nutrient pollution. And that's something that we can control at local scales. So while maybe it's harder to get a handle on what's happening at the global scale and to try to get a handle on warming, what we can do at the local scale is manage things like local nutrient pollution and try to prevent formation of these super catastrophic events like these hypoxic events. And what's becoming clear is that through this deoxygenation work, through work looking at thermal stress, ocean acidification, is that we have more and less stress tolerant species. And so what's likely going to happen is that as these stressors become amplified as we move into the future, as we have deoxygenation, we have warming, acidification, all of these factors, we're going to see a shift in reef assemblages away from the most sensitive species and towards dominance by the more stress tolerant species. And so the key here is trying to identify which species will likely do better under these future scenarios and trying to understand how the reefs are going to shift and potentially providing some insight into conservation and management or mitigation strategies. And so with that, I'd like to thank all of my collaborators. There's a lot of work here that would not have been possible without the help of students, interns, and other professors. And I am happy to take questions.
Great, thanks, Maggie. Thanks for a great talk. We have about, I'd say maybe 10, 15 minutes left. So if anyone has any questions for Maggie, you can go ahead and unmute yourself and ask, or you can drop a question into the chat and we can do questions that way.
I'm gonna stop sharing my screen now.
Okay.
There we go.
Real quick, were the fragments of the A. cervicornis and Fabiosa all from the same colony with the same genotype, or was it varied?
Very good question. So that's a part that I didn't even talk about. So we actually controlled for genotype, and we incorporated genotype as a factor into the design of the experiment. So for the Acropora experiment, we had 6 genotypes, and for the Orbicella experiment, we had 5 different genotypes. And we know that they're different genotypes because we got them through Moat's nursery and through their restoration program, where they actually know the genotypes, and they could give us like, oh, this is genotype A, this is genotype B. And so we did incorporate genotype, and we really found that there was no effect for the genotypes that we looked at, pretty much across the board. There were some that had lower baseline levels of like symbiont densities, but for the most part, their response to treatment was independent genotype. And so that's one of the reasons that we have everything pooled here. It's because genotype really didn't seem to matter. But that's a really good question, and that's one that particularly for things like coral restoration is really important, because we know some genotypes are more resilient or tolerant to different stressors. But for the ones that we use, it doesn't seem to be the case for any of those particular genotypes.
Other questions from the group?
Hi, I'm actually studying coral bleaching pathways, and I was curious as to if you know why the hypoxia is causing photoinhibition, or if there are other pathways that the hypoxia might be mediating just in the host itself, or if it's just affecting the symbiont photosynthesis.
That's a really good question, and I don't have a good answer for you. So for, if
It's
you have,
complicated, I know.
I would be open to that.
Yeah.
But what we've been doing so far is looking really at the coarse organismal scale. The work that I'm doing at Woods Hole right now is taking some of these same samples and looking at differential gene expression to try to identify why some may be doing better than others, like what mechanisms they actually have to withstand that hypoxia tolerance, or what makes them more susceptible. So hopefully I will have some sort of answer for you in the future, but as of yet, I can't answer that.
Great, thank you.
How deep were the corals in the 2017 hypoxia event? And 2, did you notice any changes in the seagrass habitat between the patch reefs in Almirante Bay?
Yeah, great questions. Okay, so how deep were the corals in the 2017 event? So for the 2017 event, we sampled the reef that was at around, let's say 3 meters, 2 to 3 meters depth. So this is what I'm calling the shallow reef. This is a reef, part of the reef that's dominated by either branching parietes or Agaricea tenuifolia. And one of the reasons to kind of specify this difference is that the shallow reef responded very differently than what I'm referring to as the deep reef, which is where we see those really massive Orbicella boulders and colonies. And the deeper reef, which is below, let's say 5 meters, is what was surveyed in the 2010 study by my postdoctoral advisor, Andrew Altieri. And so in the 2017 event, we're working with shallower corals, which may also potentially explain why they seem to be a little bit more resilient, because typically the reefs that are up in those shallow areas on a daily basis experience a lot of fluctuation in temperature, pH, potentially oxygen. And so they may already have sort of mechanisms to withstand things like hypoxic stress. In terms of the seagrass habitats, I am sorry to say that I unfortunately didn't really pay attention to the seagrass habitat. No offense to seagrass lovers. My guess is that there weren't any really big changes, because if there had been, we would have noticed them. I mean, the hypoxic event on the reef was so, how do I say, it's just mind-blowing. I've never seen anything like that on a coral reef, where like the stratification was super visible. Like we got in the water and you could see the oxygen depleted water where everything was just kind of dying. And so it's a really visually apparent process. And so if we'd seen something had been happening in the seagrass beds, we probably would have noticed that as well.
There was one other question that I didn't get to. Perhaps you already answered it, but Maria would also like to know, do Acropora and Orbicella occupy the same space or depth range on the reefs?
Yeah, good question. So it depends a little bit on what region of the bay you're talking about. And so I guess it depends on where, yes, they can. So they can occupy the same space and depths. So if we're thinking about the reefs in Panama, for example, we do see Orbicella and Acropora cervicornis in like the same sort of regions. If we were talking about Agaricea tenuifolia and Orbicella, we typically see those in different regions. And that's specific to Bocas del Toro. It could be different in some different places, but for the most part, my understanding is, at least in this habitat, Orbicella and Acropora overlap in their location on the reef.
I have a question. First of all, thank you so much, Maggie. It was a great presentation. I really enjoyed. I'm curious if you guys have looked into the microbiome, the change of the microbiome associated with under these experiments, or are you planning to look into that In particular, I'm very interested with the tissue loss and if there is some changes in the microbiome through the time series of the experiment.
Mm-hmm. Yeah, great question. So I can tell you for the field experiment we did, we surveyed the microbial community at like the benthic pelagic interface. So we took water samples, we collected the microbes, and we looked at during the event, after the event, and at the control and hypoxic site. And we did see very stark differences in the composition of the microbial community. I can't remember the names of the microbes that were more expressed at the hypoxic site, but there were some really stark differences. And that is a part of the paper that we have in review right now. I didn't talk about it because that's my co-author's specialty. But what I can also tell you is that we are sampling the coral tissue from these experiments for the microbiome. And that project is being led by Sarah Swaminathan, who's the PhD student working on the project. And she has found that there are some differences in the microbial community, but I believe she's still working on that analysis. But it's a really good question. There's definitely some pretty interesting things happening with the microbial consortium.
Fantastic, thank you.
Yeah, you're welcome.
I have a quick question for you, Maggie. You
Yeah.
mentioned that there are some co-occurring stressors with hypoxia, like acidification and increase in temperature. And I recognize those tanks at SMS.
It looks familiar.
You did a good job revamping them. So are there any plans to simultaneously manipulate some of these other parameters, or do things just get too complicated, too technical in those tanks?
There are plans for that. And so the first objective for that grant was to look at oxygen alone and see if we can see any difference between genotypes or species. Yeah. The second goal, which should have happened last summer, but we weren't able to do any experiments, was to do the multi-stressor experiments. Yeah. And that's what we're going to be doing this summer. Yeah. The plan had been to manipulate pH, temperature, and oxygen all at the same time, but I think what we're going to do is just focus on 2 to get started. And depending on how that goes, try to incorporate the other ones, because it's undoubtedly important to have those co-occurring stresses at the same time, particularly warming and deoxygenation. So I think that's gonna really change some of the results that we have.
Cool.
Yeah.
Other questions from the group?
I have one question.
Great.
Yeah, thank you for a great talk. So I've been working in Bocas for a few decades, not on the marine biology, but I don't recall ever hearing about these hypoxic events until that paper was published in 2010.
Yeah.
And the thing that struck me about the paper was that there wasn't any cause, that was mentioned. And I know that there are these conditions that are set up with eutrophication with banana plantations and sewage and pollution and oil spills and all that crud in Almirante Bay. But do you have any ideas about maybe the 2017 event, like what actually triggered it?
I have some, I guess it's speculation, but I have some potential ideas. So what could be happening is, so you're right, we have this stratification. So we know that in the bay, there is always hypoxic water now at depths below, I believe, 20 meters out in the middle of the bay. It's there year round now. So what happens when it gets warmer is, what we think happens is that zone grows and grows and grows and gets bigger. And there's no mixing, and so it just gets bigger and bigger. But then what makes that water shoal up into certain places over others, we're not really sure about. It could have something to do with some sort of like water column inversion or flipping. Like if we had a rain event and there was a whole bunch of fresh water at the surface, we could see some sort of flipping of the water column, and maybe that's pushing the hypoxic water to shoal up into different areas. It's a possibility. We really, honestly, right now, we don't really know, but I know that I believe that there are some new postdocs that are going to be working down there trying to model the area and try to actually understand what's going on, because it's pretty fascinating.
Yeah, cool. Thank you.
You're welcome.
Any last questions for Maggie from the group? All right, well, thanks everyone for joining us. And of course, Maggie, thank you so much for giving us a great seminar this afternoon. We really appreciate it. Everyone is commenting in the chat. Great talk, thank you. So we really appreciate it, and I hope to see you again soon. Maybe we'll have you back for an update.
Sounds good. Thanks so much for the invitation.
Okay, thanks everyone. Stay tuned for updates on future seminars. Bye-bye.
Bye-bye.